Hello and welcome dear students. Today we are going to discuss about the topic LOD score. The main objectives of today's lecture are to know the basics of LOD score, to discuss about parametric linkage analysis, to discuss about non-parametric linkage analysis, to understand the method of LOD score calculation, to know the facts about interpretation of LOD scores, to discuss about analysis of LOD scores for gene mapping, to discuss about human linkage analysis in the age of next generation sequencing, and finally to know the limitations of current linkage analysis programs. Dear students, let's first start with the introduction. Linkage analysis is a genetic method that searches for chromosomal segments that co-segregate with the ailment phenotype through families. It can be used to map genes for both binary and quantitative traits or characters. Linkage analysis may be either parametric, that's if we know the relationship between phenotypic and genetic similarity, or non-parametric. Parametric linkage analysis is the traditional approach whereby the probability that a gene important for a disease is linked to a genetic marker is studied through the LOD score, which means logarithm of odds, which assesses the probability that a given pedigree where the disease and the marker are co-segregating is due to the existence of linkage with a given linkage value or to chance. Non-parametric linkage analysis in turn studies the probability of an allele being identical by descent with itself. The model specifies parameters such as inheritance pattern, gene frequency, and penetrance of the disorder. Non-parametric linkage analysis seeks access evidence of allele sharing in affected relatives without a disease model. To find the disease locus, linkage analysis determines if affected relatives share a segment of DNA more often than other segments. The shared segment would be more likely to contain the altered gene. The term linkage refers to alleles being inherited together. Two loci are said to be completely linked or in complete linkage if they are always inherited together. This can be true if the distance between them is very small. A block of alleles at loci that are linked is known as haplotype. The frequency of recombination between two loci is referred to as theta or the recombination fraction. A theta of 0 indicates that the loci are completely linked, whereas a theta of 0.5 indicates independent assortment of the two loci. Linkage is measured in centimorgans, that is C capital M, with 1 cm representing 1 map unit and 1% recombination frequency. Although a cm is a genetic distance, it can be roughly related to a physical distance of approximately 106 base pairs. Recombination occurs through crossover events during meiosis, which occur as a raw material for the evolution to act upon. To find linked regions, DNA markers are used to map an area of interest or the entire genome. These markers can be VNTRs, STRs, or SNPs. To estimate linkage between markers, the number of recombinants is compared to the number of non-recombinants. The occurrence of few recombinants would indicate that the two loci are closely linked. Since it's not always possible to detect recombination due to an uninformative type of mating, the overall likelihood of the data represented in a pedigree is calculated for a set of different values for the recombination fraction. The likelihood of the pedigree at a particular recombination fraction is compared to the likelihood of random segregation. That is where theta is 0.5. A likelihood ratio presuming linkage compared with no linkage is calculated for a set of values of theta. This odds ratio is expressed in log 10 and called a Lod score, or the log of the odds of the likelihood ratio. The marker loci with the highest Lod scores are more likely to contain a segment segregating with disease. Lod scores are additive across families at each locus. A total score greater than 3 indicative of a 1000:1 odds favoring linkage meets a threshold for significance with a 5% chance of error. With the availability of polymorphic markers that span the whole genome, it is possible to scan all chromosomes for a site of probable linkage. Lod scores greater than 3 are considered to indicate regions that met further investigation, that meet genome-wide significance. Additional polymorphic markers with these regions are then tested for linkage using multi-point linkage analysis. In multipotent analysis, a putative disease locus is moved across a region spanned by multiple markers, searching for a positioning that produces a best fit to the observed frequency of recombination between the disease locus and any of the polymorphic loci. Detailed study of the haplotypes in a region of interest can further narrow the region by identification of critical crossover events. Genetic heterogeneity constitutes a major pitfall in linkage analysis. Locus heterogeneity, in which the identical clinical phenotype can be caused by defects at 2 or more different loci, can result in a subset of families having linkage between a marker and a disease, whereas another subset does not. Locus heterogeneity, therefore, can produce false negative results. This can be overcome by using large pedigree or large number of families. If there are distinct subphenotypes of a disorder, families can be stratified or classified into subclasses and tested separately for linkage if they are of sufficient size. It is also possible to perform a statistical test of admixture. This assumes that a group of families consists of 2 subsets, a proportion alpha that is linked to the locus and a proportion 1 to alpha that is not. Linkage data are analyzed for values of alpha between 0 and 1, searching for a value that produces a best fit with the data set. The Lod score method is broadly concerned with issues of power and the posterior probability of linkage, ensuring that a reported linkage has a high probability of being a true linkage. In addition, the method is sequential so that pedigrees or Lod curves may be combined from published reports to pool data for analysis. This approach has been remarkably successful for 50 years in identifying disease genes for Mendelian disorders. Dear students, let's now understand the parametric linkage analysis. Parametric linkage analysis is limited by the need to assume a genetic model and a set of parameters, but it is the most powerful mapping approach if the chosen model is correct. Since complex trait disorders by definition are not inherited in a particular Mendelian pattern, this form of analysis is not as useful in such disorders. To increase the power of linkage analysis in the face of the complex nature of common diseases, an analysis method was devised that does not necessitate the definition of parameters or models the so-called non-parametric or model-free method. This approach depends only on the assumption that two relatives affected with the same disease will share predisposing alleles, and it requires no further assumption of inheritance or recombination parameters. Although this is robust for complex trait analysis, it is also imprecise and less sensitive than parametric methods. Dear students, let's now understand the non-parametric linkage analysis. Non-parametric analysis is dependent on allele sharing. At any given locus, two people could share an allele for two different reasons. Either they have inherited the allele from a common ancestor, or the allele arose in two different populations of ancestors. Two alleles are called identical by state, IBS, if they exist in the same detectable form but are only identical by descent, that's IBD, if they were inherited from the same ancestor. Several methods have evolved around allele sharing non-parametric methods. In the affected pedigree member approach, members of multi-generational pedigrees are genotyped for multiple polymorphic loci. If affected relatives share alleles, that is IBS, more often than expected due to chance, this favors linkage. The most common application of this method is by testing affected siblings who are easier to ascertain than more distant relatives in large pedigrees. These methods use only part of the pedigrees, that is affected relatives, and are not fully non-parametric in that to extend to multi-point analysis, recombination fractions between markers need to be specified. A method called non-parametric linkage was developed to address the shortfalls of previous non-parametric approaches. The first step of a non-parametric linkage study is a multi-point analysis to extract the most data from a pedigree by establishing IBD based on a large number of markers. Since the inheritance pattern in these pedigrees is usually ambiguous, the non-parametric linkage method averages the test statistics over all possible patterns of transmission. The static is normalized and weighted across pedigrees. It establishes a probability of linkage for any point on the chromosome with the available marker information. A further extension of this method is the adaptation to utilization of discordant SIB pairs to identify areas that are co-inherited infrequently. Non-parametric linkage analysis is a useful tool for detection of chromosomal regions that may contain disease loci, although there has been considerable debate about a Lod score threshold that would represent significance in a genome-wide study. Another caution in the interpretation of the non-parametric Lod score results is that the linkage calculations are influenced by the presence of a strong gene disease association with a particular allele. Linkage analysis searches for within pedigree allele sharing, but if there is a significant allele association in the population, there also will be between pedigree sharing and the linkage result will be distorted. Dear students, let's now understand the method of Lod score calculation. Lod score, that is logarithm base 10 of odds, developed by Newton Morton, is a statistical test often used for linkage analysis in human, animal, and plant populations. The Lod score compares the likelihood of obtaining the test data if the two loci are indeed linked to the likelihood of observing the same data purely by chance. Positive Lod scores favor the presence of linkage, whereas negative Lod scores indicate that linkage is less likely. Computerized Lod score analysis is a simple way to analyze complex family pedigrees in order to determine the linkage between Mendelian traits or between a trait and a marker or two markers. The method is described in greater detail by Strachan and Reed. Briefly, it works as follows. Establish a pedigree, make a number of estimates of recombination frequency, calculate a Lod score for each estimate. The estimate with the highest Lod score will be considered the best estimate. By convention, a Lod score greater than 3 is considered evidence for linkage, as it indicates 1000 to 1 odds that the linkage being observed did not occur by chance. On the other hand, a Lod score less than -2 is considered evidence to exclude linkage. Although it is very unlikely that a Lod score of 3 would be obtained from a single pedigree, the mathematical properties of the test allow data from a number of pedigrees to be combined by summing their Lod scores. A Lod score of 3 translates to a p-value of approximately 0.05 and no multiple testing correction is required. Dear students, let's now discuss facts about interpretation of Lod scores. Number 1. Positive Lod scores suggest stronger support for theta than for half negative Lod scores, the reverse. Higher Lod scores mean stronger support, lower means the reverse. Lod scores are additive across independent pedigrees and under certain circumstances can be calculated sequentially. Fourth, for a single 2-point linkage analysis, the threshold LOD equal to 3 has become the de facto standard for establishing linkage, that is rejecting the null hypothesis of no linkage. When more than one locus or model is examined, the remark is 4 must be modified sometimes dramatically. Dear students, let's now understand the analysis of Lod scores for gene mapping. Genome-wide linkage analysis has been extremely successful at identification of the genetic variation underlying single gene disorders. However, linkage analysis has been less successful for common human diseases and other complex traits in which multiple genetic and environmental factors interact to influence disease risk. Multi-point Lod scores are typically calculated for a grid of locus positions, moving the putative disease locus across a fixed map of genetic markers. The resulting Lod score curve is then plotted against this map and the maximum Lod score is noted. Particularly given densely spaced markers, there may not be a single clearly best marker order, and the evidence for linkage may differ substantially depending on the order of a set of markers and or the distances between the markers. Changing the order of a set of markers and or the distances between the markers can make a substantial difference in the resulting multi-point Lod score curve and the location and height of its maximum. If the best order by a particular criterion, usually maximum likelihood, is enough better than the second best order, the usual approach is simply to condition analysis on the best order. If there are two orders that are nearly equal, likely one might do the analysis twice, once with each order and note any important difference in the results. If there are no important differences, the uncertainty in the order may not pose much of a problem. If, however, the two orders yield substantially different results, the uncertainty becomes a concern. If there are more than two marker maps under consideration, the situation becomes even more complicated. Researchers described a genome scan for genes contributing to type 2 diabetes susceptibility and presented an example that illustrates this problem. The data consisted of 330 Mexican American affected SIB pairs from 170 SIB ships with no parental data. Their best results occurred on chromosome 2 at the tip of the Q arm. Their maximum multi-point Lod score ranged from 2.7 to 4.3 depending on the marker order and distances they used. Dear students, let's now discuss the human linkage analysis in the age of next generation sequencing. Although linkage mapping for disease gene discovery was largely surpassed by large-scale genome-wide analysis sequencing studies due to the introduction of SNP microarrays, the era of next-generation sequencing has enabled linkage mapping to be revisited by geneticists. Combining linkage analysis results with either whole exome or whole genome sequencing results has become an extremely powerful tool for identifying disease genes. In the current genetics genomics environment, usually smaller families undergo linkage analysis using either microsatellites or SNP microarrays. The main reason for this being that many large families with clear Mendelian inheritance have already undergone linkage analysis in the previous decade and thus have data already available. Prospective simulation analysis will frequently determine that these smaller families cannot meet genome-wide significance, that's a Lod score greater than 3.3. However, Lod scores less than 2 can exclude large regions of the genome that will not harbor the causal mutation. DNA from all available family members will undergo linkage analysis. However, due to higher costs, only a few informative individuals from the pedigree will undergo next generation sequencing. Dear students, let's now understand the limitations of current linkage analysis programs. Although the introduction of computerized calculation of Lod scores provided a platform for geneticists to regularly perform disease gene mapping studies, linkage programs have computational limitations. Improvement and enhancement in the algorithms over recent decade has improved efficiency of the programs, but computational burden is the major limitation and is an ongoing problem with the current linkage programs. Computational complexity increases with an increasing number of genetic markers or pedigree in individuals. The Elston-Stewart algorithm, which is used by linkage and fast link programs, scales exponentially with the number of marker loci considered and generally cannot handle more than 5 to 10 markers at a time. This is a serious limitation in the current environment where hundreds of thousands of genetic markers can be used per person in linkage analysis. Computational time and memory requirements only increase linearly with additional markers but increase exponentially as the number of meiosis in a pedigree increases. In addition, there is an upper limit of 32 meioses in a pedigree because of the representation of inheritance vectors as 32-bit integers. To date, there is no widely used linkage analysis program that can handle both large pedigrees and a large number of markers. There is a need for more powerful methods and enhancements to gene mapping algorithms. Dear students, let's now conclude the lecture of Lod score. Linkage analysis is the method of associating the functionality of genes to their chromosomal locations by looking for evidence of co-segregating between genes and similarity in phenotypes between individuals who share alleles at those marker loci. A Lod score is a statistical estimate of the relative probability that two loci, for example, a disease-associated gene and another sequence of interest, such as a variant or another gene, are located near each other on a chromosome and are therefore likely to be inherited together. Lod score is used while mapping the relationship of genes genetically from one to another. Lod score analysis is used to estimate whether the observed degree of concordance of a genetic marker with a trait or character of interest indicates genetic linkage between the two. Lod analysis is typically used in genome-wide association studies to map a trait of interest to a particular chromosomal region. Lod score analysis requires a fully mapped genome with many thousands of genetic markers on all chromosomes and is complicated by many factors that are built into the mathematical model. A statistical estimate of whether two genetic loci are physically near enough to each other or linked on a particular chromosome that they are likely to be inherited together. A Lod score of 3 or higher is generally understood to mean that two genes are located close to each other on the chromosome. In terms of significance, a Lod score of 3 means that odds are 1000 to 1 that the two genes are linked and therefore inherited together. Linkage studies directly examine the transmission across generations of both disease phenotype and marker alleles within a known pedigree, seeking correlation that suggests that the marker is linked with a causal locus. Dear students, it was all about today's lecture regarding Lod score. Hope you have understood it well. See you next time with a different topic. Till then, take care and goodbye.